In this lab exercise, you will use chemical reactions to detect the presence of carbohydrates, proteins, and lipids. Two of these biological molecules are polymers. Recall that polymers are synthesized by dehydration reactions. The figure shows how the removal of a water molecule joins two monomers. Conversely, the addition of water in so-called hydrolysis reaction will break down polymers into monomers. Please note that for all tests, you will use test tubes that hold 10 milliliters. In those tubes, 1 centimeter equals 1 milliliter. So use the Sharpie marker to number the test tubes and to mark on each one a 1 milliliter line. For all tests, you should also wear gloves. You will first test for the presence of starch, a complex polymer of glucose monomers. Iodine is used to test for starch. When the iodine binds to starch, it changes from its amber color to a dark blue or black color. Do all of the reactions that you set up and complete table 2.1 with your results. That table is on page 28 of your lab manual. The second carbohydrate you will test is for simple sugars. You will use Benedict's reagent, which detects the presence of reducing sugars. This reagent requires heating to produce a color change. You can use a heat block or a water bath depending on availability. Please allow about 5 minutes for the reaction to take place, again in the presence of heat. The blue color of Benedict's reagent may change as indicated in this figure. Please notice what each color indicates. Once you have all of your results, please complete table 2.2 on page 29. Now you will test the presence of proteins. You will use Biuret reagent. The color change is given by the presence of peptide bonds. Recall that peptide bonds join amino acids to form polypeptides. Complete all the tests, look at the sample here, and enter your results in table 2.3 on page 30. This time we're not going to do the emulsification test, so you can skip that. And next you will do a test on available unknowns, whatever your instructor indicates. Use the same procedures you learned and complete tables 2.6 and 2.7 on page 33. For cleanup, please dispose contents of test tubes in the designated carboy, normally by the sink. Then place the empty tubes in the plastic bin that should have soapy water. Thank you.